So I guess the key things that we're learning in the microbiome gut-brain axis are, well, in two big areas. One is about mechanisms. So we're really trying to drill down into how do these microbes signal to the brain. And the other one is in terms of human translation and how translatable are these really exciting studies, largely in mice and rats. How can we translate them to humans? And so for me, that's where I see huge efforts, and that's what we're trying to really play a big role in. So it's clear that people in the areas of psychiatry and even well-being, if we want to use a more broader term, mental well-being, are those that are more likely to be exploited for clinical translation in the first instance. And so we've exciting studies having emerged in areas like mild anxiety, depression, cognitive enhancement, but these are small studies. We need a lot more of them. We need them to be longitudinal. We need to understand what strain, in what patient, at what stage in their lifespan. These are big questions that we're nowhere near yet figuring out. We need to know the relative contribution between using specific strains, combinations of strains, combining that with prebiotic approaches or dietary approaches. And I think that's where we're going to see a lot of excitement. But it requires both investment, but also collaboration between industry and academic partners to be able to move that forward. And so while a lot of the work to date has been in healthy volunteers, and we've seen across the lifespan, both from early life all the way to aging, but we need to do more in patient populations in that regard. The second part of that question then is what we're seeing very strikingly is that the neurological disciplines are beginning to really move quicker into this area than they were previously. If we have concepts that the microbes can affect the brain and that might improve psychiatric illness, then the same things are beginning to apply for neurological illness. And we can't forget that, for example, in certain epilepsies we treat by diet already. So, and diet is the biggest factor of influence our microbiome. So that's what we're going to see a lot more evidence coming out in the next few years in areas like multiple sclerosis, stroke, epilepsy, Alzheimer's disease, and Parkinson's disease. So what we're beginning to really try and understand in my lab is really to tackle the two problems that I mentioned. The problem of mechanism. So what happens if a specific microbe is in the gut? What does it do? What does it produce? And then how does it hijack the processes that we have to get to the brain? So a number of people in my lab are working on this, and we have people trying to trace using sophisticated state-of-the-art neuroscience tools to actually see how the vagus nerve, for example, is playing a role, but other mechanisms as well, like metabolites that are being produced by microbes. Can we, use them and bypass the microbes altogether? And then for the human translation, one of the key things what we're really intrigued about is the triangulation of diet, microbiome, and mental well-being. And so we know that diet affects mental well-being. We know the microbiome affects mental well-being. We know diet affects the microbiome. But really closing off that triangle is something that's really important to get at mechanistically and to understand the constraints on that within a human population who are not, having the same lifestyle as our mice in the cage. So we're really at the beginning of what we've coined as a psychobiotic journey, and we have a book called The Psychobiotic Revolution, and we're at the beginning of this revolution because it's a paradigm shift in neuroscience to be able to think that we could target the brain by targeting the microbes in the gut. And that's what this meeting is really about. It's about really showing the state-of-the-art science that's illuminating where we're going and the progress we're making. And so the revolution is just beginning.
Okay, so well, I think that one of the key takeaways is just the development of the field. So the fact that we have so many young investigators presenting both at a trainee level and at new people have set up their lab in the last decade that have emerged into this field and actually established enough to present at a meeting of this stature is impressive. So I think that one takeaway is we're attracting exceptionally talented young investigators to the field, and I think that's going to make a big difference. So my work in my home lab, because I have a couple of different hats I wear, examines how our own host genetics and the environment interact to influence the immune system and the microbiome, and how that together influences brain health and brain function, linked to things like neurodevelopmental disorders or to depression. And so I'm also connected to clinical networks that are in those domains. So I have the fortunate ability of doing mouse experiments and then going to the clinical populations and testing some of that out in those domains. So we're very interested in how those microbes talk to the immune system, particularly the adaptive immune system, and how that balance in early life influences how we respond to stress later in life, or how it might change the wiring of our brain.
So what did you think in particular about some of the clinical data that has been shared, even some unpublished data that have been shared for the past 2 days? Anything really striking in your mind?
Well, I think that it's all moving in the same direction. So we started with sort of who's there from a perspective of the bacterial taxa, and we can measure who's there using simple tools like 16S ribosomal RNA sequencing, but what we really need is to think about the function. And so I think it's emerging that things like short-chain fatty acids are influencing this both in the animal studies and in the clinical studies. People often criticize the field for not being translational, but I think the evidence shows very clearly that the stuff that was originally, or the research that was originally done in animals, is actually being reproduced in people. And so I think that we saw some evidence of that. I don't think, I think that we'll when we listen to John later, he's going to say, we don't want to overblow the field of research, but the quality of research in this area is under a lot of scrutiny, and I think that's bringing the bar up. And so we are, in fact, seeing evidence of mechanistic work in both animal studies and in clinical populations. So what I'd like to see is people in clinical populations, and even in animal studies, taking into account this individual differences component, right? Because there's so much heterogeneity in the microbiome that we're seeing population effects, but sorting that down into subtypes or different biotypes within those clinical cohorts, I think, is going to be the next step forward.
Yeah, there's been a few comments about even sex differences between male and female. So what is your point of view on that?
I'd love to know what the signature of male and female depression is. I think that's a tenable product that could come out of the microbiome. That we could start to understand why men and women are different in the way they present, the way that they respond, and the treatment response predictions.
So what can we hope for your team really in the next years in terms of discovery? What is your best hope for you and your team?
Well, I think our discovery is analyzing our data. We have so much data that this, we've learned that big data means big QC and big cleanup. So I think for us, we're moving into, adding some wrinkles to how the microbes and which components of the immune system are part of this sex individual difference component, and how does the host talk back to the bacteria. So we know the bacteria talks to the host, but how does that reply influence the microbes themselves? The American Gut Project is a project run out of Rob Knight, somebody that is still in Rob Knight's lab, but they sample communities everywhere, and I think they use the tools of that American Gut Project and that crowdsourcing thing. We've just recently initiated the Canadian Gut Project in association with them, and so that's being the pilot of that's just starting. I think the kits were actually being shipped, and so that's Halifax, Hamilton, and Vancouver have sites that are going to do a feasibility pilot for the Canadian Gut Project, and so we're all very open to that. We have funding from another source to do it. So I think in Canada, we run a lot of clinical trials, and those people, if you're funded, adding that in wouldn't be a cost prohibited as it is in some other places. So I think that you're going to see a lot more country gut projects emerging.
Yeah, that's going to change some of the research, obviously. Maybe as just a last question. So, what do you think about the event itself? How it evolved from the first edition?
Well, I think that, as I think a few people have said it, but it brings the right people together. So a lot of times in the microbiota gut axis, we end up in a meeting without people who know the things we do. So this is a nice balance of people who know both about the gut or about the microbes and the brain, and having that number of people to interact with and the trainees to get access to the people who have been doing the work is really a good opportunity. And I think it's been a fabulous event. I started from the immune brain component and added the gut brain into it when I set up my lab, right? Because I landed in a GI-rich space. That's what they were interested in. And I think when we did those first experiments looking at behavior in the germ-free mouse, we actually thought we were going to get an animal model of depression, right? We actually got reduced anxiety-like behavior. And it took several years to get buy-in from the neuroscientists. And I think Philip said it right, is will this evidence change the way we clinically treat neurology? There's a lot of steps. We have to get the public on board, we have to get the scientists on board, and we have to get the clinicians on board, and we have to get the government on board. Like, there's a lot of steps to bring this to the place where this great, fast-moving field actually has an impact on the individuals who need it.
Well, I think the key message is that I think the microbiome research and gut-brain axis research gives us really new viewpoints on neurological diseases. So there is many open questions. We don't really understand for many disorders, in particular neurodegenerative disorders such as Parkinson's and Alzheimer's, what is really at the root of the disorder. So what is the initial cause for it? And I mean, at this point, we don't know if the microbiome is the explanation for it, but at least understanding the connections and the vast impact that the gut and the gut microbiome has on brain function, brain structure, integrity, also neuroinflammation. I think this will give us new tools and better understanding of what processes are actually at play. And so hopefully we'll be able to identify new targets for treatment interventions as well. So, and I think my main take-home message is that there is already accumulating evidence in animal models and also beginning now in human studies that the microbiome and gut-brain axis are relevant for a number of diseases, for example, multiple sclerosis, epilepsy, Parkinson's disease. And so I think we have now proven the point that the microbiome gut-brain axis are important. And so now we should continue in that direction and to broaden our understanding. And I'm pretty confident that we will get better understanding and new tools to treat these diseases. Yeah, so as a neurologist, I see quite a lot of Parkinson's patients. And it actually turned out that in particular, patients that are in the early stage of Parkinson's disease, where they would not have that many movement disorders. So they're still, they don't have that many tremor, not slowness that much. So they're very independent still. But many of them suffer severely from gut symptoms, in particular constipation. And some of these patients would actually talk more about their difficult constipation in the office than they would actually talk about their other Parkinson's symptoms. And about 15 years ago, it was proposed that actually, based on pathological examinations, that the origin of Parkinson's could actually be in the gut. So it was described by Braak, which is a neuroanatomist from Frankfurt in Germany, that actually you can see in the gut of Parkinson's patients the same changes in the enteric nervous system as you can see in the brain of Parkinson's patients. And there is also a certain group of subjects where you would see those changes only in the gut, but not in the brain. And so he proposed that actually the gut could be the origin. And if you combine that with the severe symptoms of the patients, then it got me interested basically in the whole role of the gut, which is it's pretty distant from the traditional neurological field of view. So in the sense that we usually don't bother about it that much, but in Parkinson's it just was so evident. And then I learned about the new sequencing techniques that allowed the microbiome assessment pretty effectively, and so I just started to talk to people at the university who are using these methods, and I got them excited and interested in Parkinson's disease, and so then we started working on it. So and I think that's the main reason. So it started really with the patients complaining about the symptoms. I think this conference is special in the sense that it really brings together people who are interested in the nervous system, the neuroscience, brain function, and the microbiome and the gut. So usually you have conferences for each of these fields. So you have neurological conferences, neuroscience conferences, and you have like microbiome conferences. And so there's actually not that many events where these people are actually brought together. And so I think this makes this concept pretty unique so far, that it really is one of the, actually I think, main forums that brings these people together and are all really interested in the combination of these fields, or in the intersection of neuroscience and microbiome and gut physiology. I think it's a good opportunity to get updated on the progress that has been made in different research groups. And obviously there is several people that I already know and that I interact with, but there's many new faces as well. So I'm excited to see what new ideas they bring into the field.